Okay, so here we're going to go through the steps of glycolysis. And so, of course, glycolysis is going to be a mechanism for your cells to make ATP. Glycolysis only occurs in the cytoplasm. And all your cells can do this, including your red blood cell. And it is the sole source of ATP for your red blood cells. So in glycolysis, you are going to start with glucose from your diet, and then you are going to add a phosphate group through one of the hexokinase enzymes. And so there are 4 hexokinase. In the liver, this is called glucokinase or hexokinase 4. And it has a high Km, low affinity for glucose. So again, on your glucose, that number 6 carbon is going to be phosphorylated. So our anomeric carbon is number 1. And our carbon outside of the ring is carbon 6, and that's what gets phosphorylated. Now, importantly, this is not a commitment step for glycolysis, but it is a step where the cell has to do something now with this glucose. We are going to have kinases that are going to add phosphate groups. So this is hexokinase, which is doing this. And the acceptor is going to be a D-glucose. Now, some hexokinases, especially glucokinase, can also phosphorylate fructose and mannose. And in liver, you can also, and these sugars can get phosphorylated by the hexokinases, called fructokinase, for example. Now, again, this is not a commitment step. You can go on to glycolysis, or you can go into storage through glycogen. You can go on to make your 5 carbon sugars, which are needed for DNA and RNA through the pentose phosphate pathway, as well as other things. Sugars are important for many parts of cell activities. Now, the second step of glycolysis is going to be a reversible isomerization. This is where you're going to go from glucose 6-phosphate to fructose 6-phosphate. And what's important to point out here is in glucose, you have 5 of your 6 carbons that are part of the ring structure. In fructose, you only have 4 of those carbons. You have 2 carbons that are outside of the ring structure. And for glucose, it was that 6th carbon outside of the ring structure that was phosphorylated. So we're going to have to phosphorylate another carbon on the structure, and to do that, you change from glucose to fructose to free up this position 1 for phosphorylation. Now this is through an isomerase, and this requires magnesium because you do have a phosphate group there that you do need to neutralize those charges. This is a phosphohexoisomerase. Now in the third step, this requires ATP. And this is going to be a commitment to glycolysis. So this is an irreversible step that's rate limiting. Now what an irreversible step means, it does not mean we cannot undo this. It simply means that the enzyme that did this cannot undo it. In order to undo it, you need to do something else to this molecule. So in the case of reversible reactions, it's the same enzyme that can do the forward and the reverse reaction. In the case of an irreversible, the enzyme can only go in one direction. So in this case, PFK1 or phosphofructokinase 1 can only take fructose 6-phosphate with that now 1 carbon position available to be phosphorylated, and it will add a phosphate to that side. And so now we have a fructose 1,6 bisphosphate. We have a phosphate on the 6 position. We have a phosphate on the 1 position. Bis means 2, so you have 2 phosphates on this fructose molecule. So this is going to be a very regulated step. And we're going to talk about its regulation throughout the future lectures of biochemistry. Now, again, key to convention, 2 phosphate groups or 2 phosphoryl groups at 2 positions are given the term bisphosphates. Now, the fourth step of glycolysis is we have this phosphorylated compound on the 6 and 1 position, and they have 6 carbons. And so we're going to want to split this into 2 3-carbon compounds. And we do this through an aldolase step. And you're going to generate a glyceraldehyde 3-phosphate, which has 3 carbons with that phosphate on the 6th position, and a dihydroxyacetone phosphate. Again, 3 carbons, and this one had the phosphate on the 1 position. So this is DHAP, and this is glyceraldehyde 3-phosphate. It's glyceraldehyde 3-phosphate that would continue in glycolysis. The DHAP has to become glyceraldehyde 3-phosphate, and then it too can continue into glycolysis. Now, step 5 of glycolysis is the end of this preparatory phase to make actual energy. So far, we've used 2 ATP to phosphorylate our glucose, and then to phosphorylate our fructose, 1-phosphate. And so here we're going to have our interactions between our dihydroxyacetone phosphate and our glyceraldehyde 3-phosphate. And again, our glyceraldehyde 3-phosphate will continue in glycolysis. And so through an isomerase, a triose phosphate isomerase, you're going to convert your dihydroxyacetone phosphate to our glyceraldehyde 3-phosphate. Okay. So the triose phosphates and the interconversion between glyceraldehyde 3-phosphate and DHAP is important because it's only glyceraldehyde 3-phosphate that can continue. And 1 molecule of glucose will then result in 2 molecules of glyceraldehyde 3-phosphate. And the 6 glucose carbons will go through the full glycolysis. Keep in mind that 2 ATP again were used in the prep phase to phosphorylate. One was phosphorylating that 6-carbon position of glucose, and the second was going to add another phosphate to the molecule at that 1-carbon position of the fructose. Now again, we're starting here to pay off and make our energy. So in this case, we have glyceraldehyde 3-phosphate and an inorganic phosphate, and this is going to be a dehydrogenase now that's going to add your phosphate to glyceraldehyde 3-phosphate. And this is going to generate a 1,3-bisphosphoglycerate. And again, you have a phosphate on that 1 position, you have a phosphate now on that 3 position, so it's a bisphospho. And this is going to have a positive, so this is going to require coupling in order for it to get through. Also, this is going to use up NAD. NAD is going to pick up an electron and generate NADH. Now in step 7, you're going to take the 1,3-bisphosphoglycerate, and through a kinase, you're going to release 3-phosphoglycerate. And so this is the formation of your first ATP molecule. And so keep in mind that this is one of the 3 carbons coming through. When the second, third carbon comes through, it's going to generate another ATP, of course. This is going to leave you with 3-phosphoglycerate. So you still have another phosphate you can remove and generate another ATP. So as a side note, the formation of that 1,3-bisphosphoglycerate, as I pointed out, had a positive delta G. The formation of 3-phosphoglycerate and ATP has a very favorable negative delta G. Therefore, these reactions are coupled together with a net additive delta G in the minus, which means they are favorable. The products are favorable over the reactants. Now, in the payoff phase for the next is going to be through a mutase. And so here we have 3-phosphoglycerate, and we're going to generate 2-phosphoglycerate. And this is going to be through a mutase. Now, the conversion of this is not simply the mutase takes your phosphate and moves it to the position 2. What basically has to happen is you're going to have an intermediate, which is going to be a bis phosphoglycerate, and so your bisphosphoglycerate is going to be a 2,3. So basically, you're going to add a phosphate to this 2 carbon. You're not going to move the phosphate from 1 to 2. And then you're going to release the phosphate from the 1 carbon. And so that leaves behind this 2 carbon phosphate group. And again, this takes energy. It's a positive. And you can think about moving these 2 large bulky phosphates together is not that favorable. And again, we see we have magnesium to stabilize those charges. Now again, this is phosphoglycerate mutase that does this, but it brings in a second phosphate and then releases that first phosphate. Now in step 9, you're going to have that 2-phosphoglycerate, and you're going to have a dehydration of this. And then you're going to do this so that you can have a lower phosphoryl group transfer potential. And this will be transferred to phosphoenolpyruvate, or PEP. Now this is done through an enolase, and this is a reversible reaction, so we haven't yet gotten rid of our phosphate group. It's still hanging in there. And then we go to pyruvate kinase, which is where we're going to generate our next ATP. So in this case, we have our phosphoenolpyruvate, and then this through pyruvate kinase with magnesium and potassium are going to generate our 3-carbon pyruvate and another ATP molecule. And this is highly favorable. So PEP hydrolysis, or phosphoenolpyruvate hydrolysis, has a negative 61.9 kilojoule per mole of energy in that phosphoanhydride bond. And this is going to push everything towards making ATP. Now you're going to have tautomerization of your pyruvate from the enol form to the keto form. And the keto form is going to predominate at our pH, our cellular pH of 7. And this doesn't require any enzyme at all for this to happen. So in summary, glucose is going to be a universal path to get ATP for most cells. You're going to generate ATP and conserve energy in NADH to tap into later. All glycolic enzymes are in the cytosol. This takes place only in the cytosol. You are going to have 10 different intermediates, and you're going to have phosphorylated compounds of 3 or 6 carbons. The preparation phase is going to eat up 2 ATP, and you're going to convert glucose to that 1,6-bisphosphate. And then you're going to have the bonds between 3 and 4 of that 1,6-bisphosphate cleave to form those 2 triose phosphates. That's glyceraldehyde 3-phosphate and our DHAP, dihydrogen acetone phosphate. Now in the payoff phase, your glyceraldehyde 3-phosphate is the one that moves on. This is going to add a phosphate, an inorganic phosphate group to this. And then eventually you're going to generate ATP and NADH per triose phosphate that's going to be oxidized. Now glycolysis is going to be regulated, and there are key steps that are going to be regulated to make sure that you have a steady supply of ATP or you store your carbons for later use.